Good morning, everyone. Myself, Dr. P. Samuel, Assistant Professor of Biotechnology, Ayya, Nadar, Janaki, Ammal College, Sivakasi. See, in this video tutorial, we are going to see about transcription. Transcription is the very first step in gene expression. The term transcription refers to copying DNA into RNA. Copying DNA into RNA is called transcription, and it is the very first process in gene expression which is taking place in all animal cells. Let's move on into the topic. Here I am giving the basic representation of an animal cell, which is divided into two main areas. One is called the cytoplasmic region. The cytoplasmic region contains all the cellular compartments. All cellular compartments is in the cytoplasmic region, and whereas the nucleus is the most prominent region of the cell, because nucleus contains the genetic material which is said to be the blueprint of life. DNA contains all the instructions required by the cell for its function. So, inside the cell there are two distinct regions. One is cytoplasmic region, another one is nuclear region. Nuclear region containing the nucleus with densely packed DNA. Now we are focusing on the nucleus alone. See how the DNA is condensed inside the nucleus, right? So the main reason behind the condensation of nucleus is the presence of a protein called nucleosome, and still people are wondering, because when we stretch the DNA from the nucleus as a straight line, it extends up to kilometers, right? This makes the scientists to think about the wonders of DNA, right? It is such a complex molecule and such a molecule of interest, DNA, right? This is how, when the DNA is taken outside the nucleus, right? It is a very complex molecule. In the previous video tutorial, I told you that DNA is organized in the form of tangled mass, right? Here we couldn't find the start end as well as the tail end, right? So this is how the DNA looks inside the cell. Next slide. Fine. Transcription usually takes place inside the nucleus, no doubt in that. And this is in case of eukaryotes, whereas in prokaryotic organism, both transcription as well as translation taking place in the cytoplasmic region alone, because in prokaryotes there is no distinct nucleus, and hence the nuclear material arrangement is otherwise called as nucleoid, right? Whereas in animal cells, the DNA is organized in the form of chromosomes. So it is in an organized pattern. That is why it is called chromosomes. And the transcription process, it begins in the promoter box, right? What is promoter box? So promoter box has the orientation CAAT. In other words, it is called as cat box. So from the beginning of the DNA and to the end of the molecule, there are millions of bases. There are millions of nitrogenous bases like adenine, thymine, guanine and cytosine. But there are orientation like as I told you, cat box, which makes the enzyme, RNA polymerase enzyme, to recognize the promoter site. And in eukaryotic cells, the orientation of promoter site is CAAT box, whereas in prokaryotes the orientation of promoter box is TAA, Tata box, and cat box. In prokaryotes it is called Tata box, in eukaryotes it is called cat box, right? Don't forget it. Right. So in the promoter site, the RNA polymerase enzyme binds in the DNA molecule, right? So when it binds, it started to read the nitrogenous bases present in the DNA. It gradually starts to read. So when it is reading, it is copying the DNA into another form called RNA. RNA is called as ribonucleic acid. During transcription process, DNA is copied into RNA, right? So as the process continues, as the RNA polymerase enzyme complex moves, the length of the RNA also increases, right? See, it is growing, still it is growing, right? So RNA is an enzyme involved in the transcription process, no doubt in that. It starts the transcription process from the promoter site. Promoter site is nothing but the recognition site for RNA polymerase enzyme, from where the transcription process begins, right? And this newly synthesized RNA is called nascent RNA. What is nascent RNA? It is an unstable one, because for translation process, the single-stranded RNA molecule has to travel into the cytoplasmic region. During that time, there are possibilities for the digestion of RNA molecules by the host-dependent hydrolyzing enzymes. So, the nascent mRNA molecule is highly unstable. This molecule is not transported to the cytoplasmic region because the RNA may contain necessary information for protein synthesis, right? Loss of this RNA is proportional to the loss of many functions, right? So it is very, very important to protect the RNA. It is protected by the host mechanism itself. Let's see what are all the protection mechanisms. There are two mechanisms being adapted to protect the nascent mRNA molecule. So to protect it, there are two modifications are taking place. One at the 5' end, another one at the 3' end. The modification taking place at the 5' end is called addition of methyl group to the guanosine residues. It makes the host system to recognize that it belongs to our own cell, right? The second modification is at the 3' end through the addition of multiple adenine residues, and this reaction, addition of multiple adenine residues at 3' region is catalyzed by an enzyme called polyadenylate polymerase, right? See, there are two modifications, one at 5' end, another one at 3' end, and in the 5' end, the guanosine residue is methylated, right? Whereas in 3' end, multiple adenine residues are added, right? And along with the modification like 5' end modification and 3' end modification, there is another group of protein called the informosome, which are bounded to the surface of RNA molecules, right? They are bounded to the surface of RNA molecules. Now this RNA is called mature RNA. This will comes out of the nucleus through nuclear pores into the cytoplasmic region for translation, right? So it is very, very important process. Transcription is very, very important. We will discuss about translation in the next module. Thanks for watching. Stay tuned with me. Bye.